So hello everybody. Today we are honored to be in the presence of Professor Mahmoud Rouabhia. Well, Dr. Rouabhia is a full professor at the dentistry faculty of University Laval. He is a senior scientist in the field of immunology, cell biology, and microbiology. Professor Rouabia's research program involves different objectives related to the oral health and well-being. Dr. Rouabhia published over 170 peer-reviewed scientific publications. Through sustained involvement in teaching and research, Dr. Rouabhia contributes to dental student training at different levels such as master's, doctorate, and postdoctorate studies. Up to now, over 100 students benefit from Dr. Rouabia's expertise. Well, students of Professor Rouabia's are frequently among prize winners in local and international scientific conferences. And Professor Rouabia's research program is supported by grant agencies such as NIH, the CIHR, the NSARC, Le Fonds de Recherche du Québec Santé, Network for Oral and Bone Health Research, and etc. So please join me in welcoming Professor Rouabhia. Dear Professor, we are eagerly looking forward to hearing your presentation.
Thank you so much, Miriam. I'm really gladly surprised that you are presenting me here. So that's a very nice start for starting. Hello, everybody. I'm really glad to be with you today to share with you some of our works. Actually, we're not sharing everything, so I may not include the work that Miriam is doing because she's one of my PhD students, actually. And today I'm going to talk to you about something you may already heard about it, but also you may already tried it. I don't know, so you will see. And what I want to talk about is the electronic cigarettes, what is also known as nicotine delivery systems, but also we can also say it as a e-vape system. So the title is summarizing or englobing these things. So that's why I'm saying the e-vape interaction with the oral tissue and the oral pathogens. So we would say why he is really concentrated on this stuff. Simply, as you may already saw that, when we're using the electronic cigarettes or the e-vape, the first side to get that e-vape, and we will define this later on, is the oral cavity. So we're getting in contact with this strange things or friendly used things, and that first contact goes through the mouth. That's why we are concentrating on that. So how is the outline of the talk that we'll be talking? We'll be defining a little bit the electronic cigarette. I'll be explaining the concept of the aerosols. You will see why I'm concentrating on that. The prevalence of this electronic cigarette, and what is the concern, if there is any concerns, and specifically I'll be concentrated on the concerns related to the oral cavity in terms of tissue and cells on this sides, and also the oral ecosystem, and finally we'll have a very short concise conclusion about that stuff. So if we go over and say, what is an electronic cigarette? So if we want to define it in writing, we would say the electronic cigarettes or e-cigarettes is a device that heats something, and this thing is a solution containing different product with the character forming droplets, and those droplets are a source of the transportation of the nicotine. In terms of device, what is the electronic cigarette? Is a cigarette-like system. So the first person who designed this electronic cigarette, the idea is to mimic the cigarettes by itself when people are smoking, and that electronic cigarette is containing a battery for heating. And then for making things easy, we're having a switch to push on and then get heat the solution. And these solutions are linked here in what we call the cartridge or the liquid holder. And then we have the mouthpiece where the people would draw with aerosols to inhale it, and for sure we have a battery to charge this electronic cigarette. So in a general form, it refers to standard or combustible cigarettes. I was saying this is characterized by the formation of droplets to transport nicotine. So this is why we're saying these nicotine-rich droplets will be inhaled by the people, and this is the idea that to get the idea of satisfying their demand on a nicotine. Cigarettes, why people don't want, they cannot stop smoking, because there is an addiction of to nicotine, and that addiction, the electronic cigarette want to answer that question, want to satisfy that idea, and at the same time, the idea is to reduce as much as possible those harmful products we find in a combustible cigarette through the use of electronic cigarette. The electronic cigarette moved through a very large development. So at the beginning, we're having a real shape of a cigarette, but this one is for a reduced time of use. So people will have their always their packets. They use this cigarette, and there is no more liquid in it, so it gets thrown out. Then things moved out to try to make it friendly, useful, not always having a new one and try to lining, there is no more liquid and go on. So it went through what we call the second generation with a small reservoir for that liquid. Then people claimed, oh, the reservoir is not so good. It doesn't take too much liquid, so I'm always trying to fill that. So we moved out to the third generation with a little big reservoir. Then the third and the fourth generation, basically the fourth, this one with a big reservoir and a high capacity battery for heating. Don't forget that battery is here to heat that liquid. We will define this later on, and then transform that to aerosol or vape like smoke. And then the last fourth generation, I would call it the fifth generation, is having that electronic cigarette as small as possible. It's like the USB we're using with our computers. And this is make it very easy to hide. And that's why actually when we see people, basically young student using this, it's easy to hide. It's in her hands, and he get his puff of the nicotine, and then no one knows that he has that things in his hand. Now this is the evolution of the electronic cigarette. And then I was talking about a liquid. As you can see here, there is a whole bunch of bottles, all of them colored, different colors to make them very attractive. And these bottles contains the different type of liquid. And these liquid, when they are heated, usually at a higher temperature, over 60 degrees Celsius, the liquid contained on these bottle that we put in our electronic cigarette is like this. It will form this type of smoke. But in reality, this is not a real smoke. I will get back to that. It's a sort of aerosols. Why we're talking about aerosol? Because it's like a humid smoke containing a different droplet rich in nicotines. And now those aerosols will be, of course, inhaled. So the basic aims of the electronic cigarette is to emit the gestures and the nicotine supply by the cigarette without combustions. The idea is to avoid burning the cigarettes, and by using that burned cigarette, there is more chemicals getting to the mouth, but also to the body. And the idea by using the electronic cigarette to reduce as much as possible these chemicals. So that idea, there is more popularity for the electronic cigarettes. People saying, I'm having something that is better than cigarettes. That's why there is an increased prevalence of the electronic cigarette. If we look at in this prevalence in Canada, there is the last survey made in Canada on 21-22, there is 18% of students in the grade of 7 to 9 had already tried electronic cigarette. If I come back and remind you why electronic cigarette is there, electronic cigarette is here usually or normally they get in to help those hard smokers to reduce the damaging effect of cigarette or combustible cigarettes, and also maybe to help them quitting. But when we look at the prevalence now, we're finding people in the grade of 7 to 9 had already tried electronic cigarette. There's 41% of the students in grade 10 to 12 reported having done so. So it is normally this population, they are not smokers. And when the electronic cigarette showed up, they tried that stuff. So when we move on and raffinate the analysis, from this population, 61% who had vaped in the last 30 days have never smoked a single cigarette. So in each way, the electronic cigarette is a sort of attraction to the young people to try it. That's one of the big issue that we're concerned about it. As I said, so electronic cigarettes were designed to help smokers to reduce and quitting. But the idea, when we look at the reality in this previous survey that I showed you, is not the idea, is not that. It is encouraging young people to use this electronic cigarette. So the purpose is, this is due to what? To the promotion of vaping as a healthier, if I vape, I use cigarette smoke, it's better than smoking. And the other idea is there is a modern way to smoke, likely plays a role in the interest of youth and young adult in using electronic cigarette. So that is a big deal. So if I get back, and this is probably only one comparative slides, is electronic cigarette safer? Of course, if I compare it to combustible cigarette, there is a big difference in terms of chemicals, but it's not the absence of chemicals, the dangerous chemical completely from electronic cigarette. So what we're going to do now is comparing the users or the effect of electronic cigarette to those who are not using electronic cigarette in terms of experiments. So the concern specifically is due to what? There is two major concerns. First of all, look at these small electronic cigarette available. As I said, you can hide them in your hand with nothing. These small ones, they are containing a high amount of nicotine. All these pods, they are going from 40 milligrams to 50 and to 90 milligrams of nicotine, which are really a high amount of nicotine. As a comparison, in one cigarette, combustible cigarettes, there is about 10 milligrams of nicotine. You look at this pulse is getting over 4 cigarettes. This is over 5, and this is over 9 cigarettes. And another concern is the maximum of concentration recommended by Health Canada in the regulation is 20 milligram per mil. These pods are containing much higher amount, and they are available to the majority of people to get it. So one saying that to use that electronic cigarette, we need a liquid on those bottles. What is the constitution of these liquids? So if we look at them, the composition of liquid, there is about 0 to 5% of alcohol, a very low amount of water, because people earlier, and I will get back to this later, people are thinking that it is a water or a nicotine-rich water. No, it is not that. Why? Because the water won't give all that smoke-like vapor there. There is 80 to 90% of propylene glycol or glycerin, of vegetable glycerin. These are the humectant. These are the two major constituents to transport the nicotine from in form of droplet and bring them to the mouth and also to the rest of the body. Of course, there is nicotine. There is some liquid that doesn't contain nicotine. And the major attractive system for young people is these these flavors. The flavors is we goes from banana to strawberry to whatever you want to get, you can get it that flavors. So that is this composition is before you heat the e-liquid. I told you there is a battery to bring that e-liquid, transform it to vapor. So when we get that e-liquid changed or heated, we're not getting a vapor as you are cooking your pasta. We're not getting the same thing as the smoke for a cigarette. We're getting all this aerosol, as I said, which is a group of droplets transported by the humectant. I remember the first time I published a paper on 17, and it was the big time where people are talking about electronic cigarette. There is one on TV who are asking, "Well, Dr. Rabi, we're talking about the danger. This is a water heated liquid, and it's not." I said, you don't know really what that, because the water never gives vape like this. And it's simply the vapor constituent water. And when you go and look to these aerosol, you're touching or you're looking at different droplet rich in a nicotine. So there was a misunderstanding of the constituent of the electronic cigarette. So if we go and look at those droplets in a microscope, so what I did, I vapor the e-cigarette on slides. And when you look at them on the microscope, look at these different droplets. It's like oil mixed to water, and then it gives you this structure. So this is nicotine free, and this is nicotine rich, meaning that when we vape that electronic cigarette, we have a complete layer of these droplets covering all the mouth, tissue, teeth, whatever, and then getting to the other airways. So these chemicals, I showed you what is in the liquid before heating, and now this is the chemical in the e-liquid after heating, meaning the aerosols. There is a whole bunch of chemicals, and those highlighted are those chemicals known to be problematic, causing even cancers wherever we use them. So if I compare it to combustible cigarette, there is low chemicals in electronic cigarette, but it's not safer, it's not free of chemicals that may cause dangers, like what I'm highlighting here. And that is one of the major concerns about the electronic cigarette. So when we get that electronic cigarette, as I said, or we vape, the aerosol, first of all, so we will heat it through that battery. This is the cartridge where it is, heat it, and then the aerosol get formed. It will be inhaled, reach the length, all parts of the body, of course, getting through the mouth, and then what left, it will be absorbed through the mouth, but also the nose, meaning that all airways are exposed to that electronic cigarette. We did some studies on the bronchial airways, some study on nose, some study on the mouth. Today I'm simply concentrating on those studies with the mouth to see the ecosystem, but also the cells. And you can find all those studies in terms of all the airways available. So what is the effect of these aerosols on our cavity? We did it in a different pathology that may increase or may promote it in the oral cavity, and one of them is the periodontitis. This is the healthy conditions of your tooth. You have the tooth is here, and here all the surrounding tissue to support and keep your tooth in a healthy situation, meaning there is a bone on this side and you have the gum on this side, and of course your tooth is here. When we are using cigarette, electronic cigarette, so we're exposing all the total of these tissue to those aerosols, and that exposure to the aerosols would leads, and there is a lot of study showing that the exposure to electronic cigarette may increase the possibility to having periodontitis. So what is that periodontitis? Look at the shape and the type of the tissue in the periodontitis system compared to the normal. It is getting inflammatories. It's getting separated. The teeth, the tooth is getting separated a little bit from the supporting tissue there. And if we look at the mouth clearly of the patient, you look at these, finally look at these, the red conditions here, meaning that the inflammation is increasing, and there is also a starting of a bleeding situations due to the deposition of all these aerosols on all this tissue there. So if I look at the condition in periodontitis, there is two big ways. As I said, in the oral cavity, you have your tissue, but you have also the ecosystem, meaning all the microorganisms we can find in the mouth. I don't want to scare you, but up to now, we identified more than 700 types of microorganisms that can be present in our mouth. Fortunately, because we're brushing, we're cleaning, and so on, it's not always a big deal. So there is a first idea on there is the disruption of the oral microorganism ecology. Why? Because when we're getting to those droplets, as I said, you have the flavors, you have the different chemicals, you heat all these, so they get into the mouth and disturbs all those ecosystem first. And in a second way, what it can do, it disturbs also what we call it the gingival innate immunity. So that innate immunity is the system that protects us at first step, and then if there is anything that cannot be solved by innate immunity, we will have our immune system in general to prevent. So in terms of the ecosystem up to now, we know that the electronic cigarette disturbs different microorganisms available in our oral cavity. We worked actually in two basic microorganisms present in the oral cavity, meaning S mutans causing caries, but also Candida albicans that causing candidiasis. So I'll be talking specifically on this fungals and interaction with the oral, electronic cigarettes. So the effect of electronic cigarettes on the behavior of Candida albicans, we know that this fungus can be present in two ways. You have it as a form of yeast, which is those round forms, and depending on the condition that you gave to these cells, they can change of forms and adopt what we call the hyphae forms. And these hyphae forms do have a more possibility to be invasive, so causing more diseases to the oral cavity, but also to the body. And once these get activated and to get pathogenic, we reach and we ended with what we call it candidiasis. The oral candidiasis, there is also skin candidiasis, there is different forms of candidiasis. I'll be talking specifically on the oral of this one. So what we did the first time is to see whether electronic cigarette can modulate the growth of Candida albicans. What does this mean? We put Candida albicans in a culture, and then we expose this different times to controls, nicotine-free and nicotine-rich aerosols. And when you look at this figure, it is even without nicotine, there is an increase of the Candida albicans growth compared to the control. And once you add the nicotine or use an aerosol that contains nicotine, there is a significant increase between the control and the exposure, and even between the nicotine-free and nicotine-rich exposure aerosols. This is one of the first steps showing that there is a clear interaction between the aerosols of electronic cigarettes and the growth and the possible pathogenicity of Candida albicans. There is a second way, as I said, Candida albicans get form changing. And it goes from the round forms to the elongated forms or elongated shapes. And once again, if we look at the compare control to nicotine-free and nicotine-rich, what you can see there is a longer forms of the hyphae when you expose the cells to the nicotine-free, but also to nicotine-rich with a higher increase with nicotine-rich. So it is two major points: increasing the growth, but also increasing the form changing, what we call it the transition from yeast to hyphae forms. And the fact that we have these two responses to electronic cigarette would say that there is a possible increase of the Candida pathogenicity when we are using electronic cigarette. Why are we interested on that? Because we know there is at least 70% of the world population are having Candida albicans in their mouth, but it is in a commensal form. And when you get it activated, it may be transformed to a pathogenic form. We also looked at, because there is an increase of growth and transition, we know that growth and transition is under the control of different genes. And these genes on Candida albicans are organized in a sort of families. And one of the family of the genes is what we call it the secreted aspartic proteases. And these secreted aspartic proteases, basically SAP2, 3, and 9, are involved in the growth, but also in the transition and in the interaction with the tissue or the mucosa. So how these genes behave when we expose Candida albicans to aerosols? And the answer is in these three figures. We're talking first of all about SAP2, and what we see, there is an increase of the expression of these genes. We did this by RT-PCR. There is actually in the three different genes, there is an increase of the expression of these SAP genes. The fact there is that increase of these three SAP genes would consolidate the idea that we have a growth and a transition promoted, both of them, by the exposure to electronic cigarettes. So this may leads to a deregulation of the innate immunity. Why? Because normally the Candida is in a commensal form. We have our innate immunity to be protected. So the idea is the fact that there is a change on the behavior of Candida albicans when exposed to aerosol would lead to change the innate immunity. That's why what we did is to look at the interaction of the electronic cigarette or the gingival epithelial cells with the aerosols from electronic cigarette. And this is what we look on. As I said, I showed you already there is an inflammation. So the tissue is like this, and then the epithelial cells are on this side, in this step. And then when we expose them, they may have a complete changes of the role of these cells there. So what we did, we isolate the normal, healthy gingival epithelial cells from donor. When you isolate these and you culture them, you will have this shape. They're cuboidal cells organized, and when you get them confluent or covered, all the space where we stop. So when you expose these cells to the electronic cigarettes, whether at 2 times or at 3 times, look at the shape of these cells, how they. These are small ones, and these are start to be big ones. elongated ones. And when we expose them to 3 times, and this exposure is only 10 minutes, 10 minutes, 3 times, once a day, second day and the third day, and how the shape is completely changing. Meaning there is a real deregulation of the structure of these cells, and meaning also there is a deregulation of the tissue in general. I'll get back to it later. So because these cells showing this shape, meaning they are stressed. A cells being stressed would release one of the enzymes well known to expose that is what we call it the lactate dehydrogenases. And we're able to measure that activity. And when you look at the control, we have this level. With electronic cigarette or aerosol, you have almost a double of that level of lactate dehydrogenases. Once again, when you look at if there is an increase, if I get back to my slide here, this shape tells me that cells are not normal, whatever after 2 exposure or 3 exposure. If they are not normal, probably they will move on to cell death types. So what we did, we looked at the apoptosis of these cells using the labeling marker, what we call it annexin positive cells, and look at the percentage or the level of apoptotic cells in the e-cigarette exposed culture compared to the control. So meaning there is an increase of cell apoptosis, or what we call it cell death, when we expose the cells to electronic cigarette. How, what is the mechanism that leads to that? We went and looked at one of the key enzymes involved in that apoptosis, what we call it caspase 3. And when you look at that caspase 3, there is a real increase of that caspase or protein enzymes there when we expose our cells. So if I get a summary of this, you expose the Candida albicans to the e-cigarette, there is a growth, transition, and virulent gene expression. You expose the cells, gingival cells, you will have a change of forms, you will have an increase of cell death through a mechanism, through a apoptotic mechanism. When we use electronic cigarette, we're using with this in our mouth. So both of cell types, Candida and cells, are together on the same sites. So that's what we'll be looking to later on. So to be sure, you may say you're using a monolayer culture. The cells are not that strong there. What we did the same work, but in a 3-dimensional engineered human oral mucosa. It's like normal tissue, normal mucosa. So this is the engineered mucosa where we have the epithelium, a stratified tissue, the connective tissue. This is non-exposed. When you expose this tissue, look at the form changing. I'm moving from 5 to 6 or 7 layers to almost only 2 layers, whatever after 4 or 8 time exposure. So there is also that the damaging effect we're seeing in monolayer is also available in the tissue, the complete tissue, confirming the damaging effect of electronic cigarette on that. Those damaging effect goes also through an increase of the inflammatory responses. And this slide really explains the previous photo I am showing you with the mouth, a completely red one, confirming that the inflammation. And look at the increased cytokines involved in the inflammation. All of these are pro-inflammatory, meaning they increase it, they promotes, they activates what we're having as an inflammation, and all of them are increased when we expose our tissue or our cells to the aerosols coming from the electronic cigarettes. Now what we did is to bring both of them, the cells and the fungus, to see the interaction and expose them to electronic cigarette. I have here not available, really easy to see the epithelial cells where I cultured also Candida albicans. Look at the shape. Yes, they are transforming, getting elongated. But when I look at the nicotine-free exposed, there is all clumps of Candida albicans growing and attaching to the culture. And when we expose this to nicotine-rich, it is more clumps and highly attached to each other, and increasing the attachment of the Candida and changing form to the culture. So when I separate or use the epithelial cell differentiation, this confirms to me what I already showed previously. When I discard all these Candida growth there, what I see here in the control, I have very few differentiated cells, meaning bigger shape cells, and compare it to the nicotine-free, where I do have more of that, and to the nicotine-rich, where there is a higher number of differentiated cells there. So meaning that I do have that the fact that we expose or use it in our oral cavity, if we are porter to the Candida albicans, this may increase the pathogenicity. So what we did also, we took those culture from here and we evaluated the viability. And as you can see here, there is a decrease of cell viability in the nicotine-rich, a little bit in the nicotine-free, but basically on nicotine-rich when the cells were cultured on the presence of Candida albicans being exposed to electronic cigarettes. That effect is also confirmed by that famous enzyme I was talking about earlier, which is the lactate dehydrogenase, is confirming the effect of aerosols on promoting Candida albicans pathogenesis. Now this is we end with all this. If I make a very small but a really important summary, the electronic cigarette may be better than a standard cigarette, but and you know the other words, meaning that electronic cigarette by itself is not that safe as we think. And now as a perspective, you may also heard that people are trying to add to these electronic cigarette some other chemicals like cannabis chemicals, meaning THC or CBD, and they are thinking, oh, it's a better way to use that way. And we already realized that the use of cannabis product with electronic cigarette is a major concerns, once again, with young people than with those hard smokers. So with this, I would like to thank all people, collaborators involved in their studies, students also involved in the study, and basically the funding agency for this research or these results I already presented. And thank you for your attention, and I'm here to answer your question if possible. I don't know, who knows?
Thank you, Dr. Rouabhia. I think usually well, we do a little bit of questions and then we take a little break and you fellows talk with Dr. Rouabhia, right?
I'm here.
So any questions? Or you want to wait maybe after?
Don't tell me everything was clear.
Well, I have some questions, but I'll keep them for the session with the fellows.
Okay, perfect. François, merci.
Maybe I have one question. Thank you for your presentation. I was wondering if what is bad in cigarette is also nicotine, or there are also other compounds that, and when the kind of experiments you showed with e-vape, when they were done with cigarette products, was it nicotine the molecule, or there's also other things that are specific to cigarettes that are pretty bad?
For cigarettes by itself, as I said, first of all, depending where the cigarette is sold. If you go, for example, and I was really surprised negatively, it looks like when you go, for example, to Africa and you buy those cigarettes, they contain more tars, what we call it goudron, and they contain more chemicals. The company are sort of allowed to include that to make it more addictive. And it looks like when we come here in terms of North America or so on, the cigarettes are under a strict control. So the companies won't include those tiny, dangerous products. And what we specifically are taking care of it, or what is present on this, is those acetyls or those formamils, they are really cancerogens. And these, basically, the amount of these products are highly present on cigarettes compared to electronic cigarettes. And depending where we are in the world, we are exposed to more or less of these chemicals.
Thank you.
Anyone else?
Yeah. I'm going to keep some questions for the fellows, but I had one more general, and maybe this is a naive question, but do we find, like, in hospitals and like epidemiological data that e-cigarette smokers have higher rates of oral candidiasis, or is there not really any numbers for that?
There is to be clear with you, there is two sort of clinical studies, and these two clinical studies, I think it appeared one on 18 and one at 20 or 21, where they did a sort of a survey in terms of candidiasis, and it looks like there is a higher level of candidiasis or candida infections, oral candida infections in people using electronic cigarette compared to those non-smokers. I'm not comparing to smokers, because smokers is already shown that there is a higher amount on that.
Okay, thank you.
You're welcome.
Thank you. I would say we could take a little break, and then you can come back for the talk with the fellows. Is it all right? Okay, see you in a couple of minutes.
See
Thank
you.
you.
Bye bye. Thank you.
Camera is still on.
Huh?
Your camera is still on. Your camera is on.
What are they doing now?
I don't know.
Yeah, I thought
It's a break.
they do have some questions, so we are going to eat and get back.
Monsieur Rouabhia, oui, c'est ça.
Oh,
Je vous entends. Je vous entendais demander, c'est ça, normalement, ils prennent toujours une petite pause de 5 minutes pour
Oh, oui.
permettre au conférencier de reprendre son souffle, puis après ça, les gens viennent vous
Ah
poser
oui,
des
non, il n'y a
questions.
y a pas de problème.
OK. Ça, je voulais juste être sûre que vous sachiez.
Ouais.
C'est François là, qui va piloter la discussion là, entre vous. Moi, je vais être cachée en arrière, puis s'il y a un problème, ben, je réapparaîtrai au besoin.
D'accord.
Fait que François, je te laisse prendre le relais depuis de là, d'accord?
Parfait. Je pense qu'on peut attendre encore une petite minute ou deux